Hello everyone, welcome to my channel. Today we shall be learning about the subject biology, and we shall be learning about the chapter biotechnology, principles and processes. First, let us know what is meant by the term biotechnology. So, first we shall know what is biotechnology. So, biotechnology, it is basically the sum total of the cells, organisms, then substituents, the natural parts of their parts, molecular analogues, etc. So, basically, the term biotechnology refers to the products, the cells, the parts of their parts, which are involved to make our life better. So, what is biotechnology? It is the parts of cells, parts of organisms, and parts of products, cells, including genes, gene products, etc. So what do we get here in this product? What does it help? It helps to modify the human health and environment. So, helps modify human health and environment. This is all about the definition of biotechnology according to the European Federation. Okay, so now let us come to know about what do we understand or what are the principles of biotechnology. Before that, let us know what are a few examples of biotechnology. It is used in vitro fertilization for the production of test tube baby. So as you know, what is test tube baby? Test tube baby means when we are injecting the sperm and the egg cell outside the uterus, we are fertilizing it, we are making a fertilized ovum, and then we are injecting it into the uterus of the healthy female, and we are impregnating it, and the baby born out of it is known as test tube baby, and this method is known as in vitro fertilization. So this also uses the technique of biotechnology. Second is we get the product human insulin, which is also known as humulin. So what do? We extract the insulin from a healthy human and we multiply it, that is we integrate the gene of insulin with the vector, that is E. coli plasmid, and then we multiply it using gene cloning using the PCR technique, and we get the product which is known as human insulin, which is a recombinant product used for various purposes, especially in people who suffer from diabetes. We inject them with human insulin so that they are able to balance the levels of insulin in their body. So, second example is human insulin or humulin. Another example which we usually see is hepatitis B vaccine. So hepatitis B vaccine, earlier it used to be isolated from the patient who used to suffer from hepatitis B. But now what we do, we are just extracting the hepatitis B gene, and we are integrating it with a plasmid, and we are growing it in multiple amounts by using gene cloning technique by the way of PCR, and we are integrating it into people who suffer from hepatitis B and hence eliminating their disease. So, second example, hepatitis B vaccine. Fourth example, we see in case of golden rice. The golden rice contains the high amounts of vitamin E because of presence of beta carotene. Beta carotene is an example of substance which is a very high, which contains a very high concentration of vitamin E. So hence, beta carotene which is present in the genome, or beta carotene which is present in golden rice, it helps to balance the levels of vitamin A in our body. So patients who are suffering from vitamin A deficiency, they can integrate golden rice in their diet. Also, for correcting or making any change in a defective gene, we also use biotechnology. So now let us come to know what are the principles of biotechnology. Also, firstly, uh, under the principles of biotechnology comes genetic engineering, and second is chemical engineering. Under principles we have genetic engineering. So what is genetic engineering? So according to genetic engineering, these are the techniques which are used to alter the genetic material or the DNA or RNA of organism in order to change its gene and to make a new recombinant product or new recombinant organism. This method is known as genetic engineering. So here the gene, the genetic material, that is DNA or RNA, is altered. So when the genetic material, that is DNA or RNA, when it is altered, we get a new recombinant organism or a new product, which is recombinant product. So we are using the rDNA technology, that is the recombinant DNA technology. So over here, the material which we are, the new organism or the new product is which is obtained is known as the recombinant DNA product, or whatever change we are causing. So it will also cause the change in the genotype and phenotype of the organism. So it causes a change in the, especially in the phenotype of organism. So how we are doing it? Let us see a diagrammatic representation of this. So this is gene A, and this is a product of gene B. So on integrating this, we will get the difference we will deeply cover it. So once we are integrating it, we are getting recombinant DNA. Now, after we are getting the recombinant DNA, we can use recombinant DNA. We can do gene cloning in order to get multiple copies of this recombinant DNA. So, after this, we do gene cloning, get multiple copies. So, why do we need multiple copies of our DNA? Think of it, for example, you are going to buy a phone in the market. So you first ask your friends which is the better phone. So you know that either Samsung or Vivo is a better phone. So you go and you ask for the latest version of Samsung phone or Vivo phone. Now, what happens is, if the latest version, if it is present only one number one, that is only one quantity is present. So it will be very difficult to give the product to all the customers. So for that, multiple copies of the same product of Samsung company phone or Vivo company phone is produced in the market so as to make it available to all the people in the market who wants to buy that same product. Similarly, here also, when we are doing multiple copies, when we are cloning the RDNA and we are producing multiple copies of the same RDNA, we are making it available to people in the market who may be in need of this RDNA in order to utilize it for various functions. That is why we are doing gene cloning with the help of PCR, this polymerization chain reaction. Now let us know what is chemical engineering. This is the second principle of recombinant DNA technology or biotechnology. What is chemical engineering? In chemical engineering, what we do, we are utilizing that is the microorganism which is being used over here will be free from contamination of microbes. So here we will take, for example, we will take a glass and we will take the microorganism which we want to transform. So we will keep it contamination free so that there is no competition in between another microbe which is going faster than the microbe which we want to transform. So here it is the contamination free zone, and we are using the technique chemicals are being used in order to transform the gene, and we are getting the new recombinant DNA product. So we have to use a contamination free zone. The contamination-free microbe zone is being utilized here, and we are using chemicals in order to produce a new recombinant DNA. So, this is the second principle of biotechnology. Thank you for today. This will be it. I hope you enjoyed the class. Please go through the topics, and if you have any doubts, ask me later. Thank you.